So, as the title of the talk suggests, I'm going to talk to you about the mitotic checkpoint and the role of the kinetochore in regulating this checkpoint. So the mitotic checkpoint is also called the spindle assembly checkpoint, and it's the mechanism to ensure accurate chromosome segregation. And this checkpoint is regulated at the kinetochore, which in yeast, budding yeast, it's 16 kinetochores that form one foci in the cell, as you can see in yellow here. So when kinetochores are unattached to the spindle, it recruits MAT1 and MAT2, the checkpoint proteins, to the unattached kinetochores, and that is what activates the checkpoint. So, what we want to ask is which proteins can achieve this arrest, and we initially want to ask which kinetochore proteins achieve this arrest, but because we have the whole yeast proteome tagged with GFP, and as you remember from Peter yesterday, we do this screen where we can fuse a protein of interest to the whole GFP library. And to show this works to recruit MAT2 to the kinetochore, we have this the GFP binding protein, which is derived from a howler monkey. We fuse that to MAT2, and we show here where we have the kinetochore tagged with GFP and the MAT2 tagged with the GFP binding protein. We can see that it's co-localized in the cell. The proteome-wide data from this screen looks like most of the screens we've done. Most of the strains don't care to have extra protein stuck to it, but we're interested in the interactions that cause a growth defect, because the checkpoint stops growth. So to confirm those top hits, we retested the approximately top 200 synthetic physical interactions. We tested those, and we validated 37, which are shown here in this network diagram. And what we found when we do a geo-enrichment analysis, the nuclear transport and checkpoint and kinetochore genes are enriched. And because we're interested in the kinetochore, we want to look at this in more detail, and especially the strongest hit, which is CEC4, which is a nucleosome that replaces a histone at the centromere. So, because of our system, it stops growth. We can't look at it unless we have a conditional expression of our fusion. So we created a construct where we fuse MAT2 to CSC4 directly, the two open reading frames, and we created the constructs under the control of the GAL promoter. So on galactose, this fusion is expressed, but on glucose it isn't. So we show here that when we express the fusion, growth is inhibited. So there is a possibility that we are just affecting the growth, affecting the essential function of CSC4 by fusing it to another protein. So to confirm that what we are doing is activating a checkpoint, we deleted the other members of the checkpoint, of the mitotic checkpoint. And we can see when we delete MAT1 and MAT3, for example, we rescue the growth defect of the fusion. But one of the main one of the key functions of the checkpoint is to prevent the cleavage of cohesin, which holds the sister chromatids together. We want to check that as well. So here we have the cohesin subunit SMC1 with YFP and histone tagged with CFP to mark the nucleus. And you should see an accumulation or stabilization of cohesin when you are activating the checkpoint, and that's what we see in when we express the fusion compared to controls. And this is a quantification of that. So just to summarize, we've shown that MAT2 activates the mitotic checkpoint, and we've shown that MAT2 blocks growth when it's recruited to the nuclear pore, and we have a tool, MAT2 CSC4 fusion, to arrest cells prior to anaphase. And this new system, the synthetic physical interaction, constitutes a new type of large-scale genomics data that complements genetics and physical interaction data. So I just want to thank my lab, Peter Thorpe, and my thesis committee, these people for reagents and high throughput genomics, and Singer Instruments for the brilliant robot.